Hello, welcome for this presentation. My name is Lukas Drag, and I'm a member of the team of Lukas Drag, mostly focusing on the woodland ecology and the conservation of saproxylic beetles. However, today I would like to talk about something slightly different. I would like to present you a brief summary of my data that I gathered and analyzed during my stay in Germany as a postdoc, under the supervision of Pierre Miller. It was a part of the Bio as Health project that was focused on the scenario biodiversity and ecosystem services. So maybe just what the presentation will be about. From the title, it's quite obvious that it will be probably connected with some forest structure, saproxylic beetles, and mostly about their traits. So let's start with some general introduction. So probably you already know that the ecological communities that exist on the different spatial scales are influenced by many different processes. The most important ones are probably the environmental filtering, which are the specific habitat conditions, the climate or the topography of the places. Then it is the biotic filtering, which are the co-occurrences and associations with different species. And also the dispersal, which means if the species can even reach those sites. It is also very well known that all these biotic and abiotic processes are influenced or might be influenced by the species traits. However, the direct relationships between the abiotic factors and these functional traits have been very rarely studied, especially for the arthropods and insects in general. This is quite important task, especially if we want to know how these traits can change in the changing environment. So as a model group, we decided to use the saproxylic beetles. It is a group associated with death or decaying wood, during some of part of their life cycle, which makes them directly connected with the dead wood. Also, these species were very commonly used as an indicators of the forest biodiversity. This is a group which is taxonomically, phylogenetically, and also functionally very diverse, including many species with different morphological adaptations or different life strategies. Also, it is quite abundant group with many species across Europe, and not only in Europe, of course, with the individuals that have very important ecological functions. For example, they participate on the wood decomposition. So what were the questions, or what is the structure of this presentation, basically? Firstly, I would like to talk a little bit about the different traits of saproxylic beetles that are available and which of them are relevant to choose for the functional analysis. Then I would like to say something about the results of those traits that we chose, and if they can predict the variation in beetles' responses to the environmental characteristics and variables. And at the end, I would just briefly show some results about if those patterns are consistent across the different forests in Europe. So let's start with some traits. As I already mentioned, saproxylic beetles are quite abundant. However, they are not very well studied, with just a few exceptions of large, mostly very famous and endangered species. For the majority of saproxylic beetles, we don't have much information about their biology, and thus all the trait-based studies are limited to only a few traits that are available. For the simplicity, I divided the traits into 3 different categories, like life history traits, ecological traits, and morphological traits. Those life history traits usually consist of some seasonality or phenology of species, and they are not very commonly used in the analysis. On the other side, the ecological traits are probably the most commonly used and very frequently in all the analysis. they consist mostly about some habitat preferences of the beetles, which means if the beetles prefer different canopy openness, wood decay stage of trees, wood diameter, or different tree species. Or there are ecological traits focusing on the foraging guilds, meaning if the beetles are, I don't know, feeding on fungi, wood, or if they are predators, or this kind of classification. It is important to note here that these ecological traits, since they cannot be measured on a single individual, but they are mostly based on some expert judgment and their opinions, very oftenly very subjective. These traits shouldn't be considered as the traits in the strict sense of way. Unlike ecological traits, the morphological traits are replicable and measurable, usually focusing on different body parts of the beetles and following some standardized protocols. However, these morphological traits haven't been used very oftenly. If so, the studies were restricted only to body size or body mass, usually, because all other traits were mostly missing. But in theory, those morphological measurements should be linked somehow to ecological functions and then also to the specific habitat conditions. At least this is known from other groups of arthropods. For example, we know that the length of mandibles could be related to the different food resources, or that the coloration of individuals might be related to their thermoregulation or to their UV protection, or very often it is mentioned that the size of the wing might be related to the dispersal ability. So we try to somehow fill this gap in this morphological traits, and we gathered quite a large data set consisting of almost 1,200 species of beetles coming from the Central and Northern Europe. And we measured them for 32 different traits, plus 5 additional traits were counted, like the wing aspect, that was the wing growth, for example, that was the proportion of the body mass and the wing area. Although we acknowledge the intraspecific variability that may exist in those, unfortunately, most of the individuals were measured only on one specimen, because otherwise this would be too much time consuming. Similarly, we kind of neglected the possible sexual dimorphism, which might be present for certain species. For example, this Lucanocerus. Okay, some overview. Not very surprisingly, most of these morphological traits we measured were highly correlated with the body size. So we decided to remove this effect of body size by extracting the residuals from phylogenetic generalized least square regression model and plotted the new correlation metrics. After accounting for the body size, we can see that we distinguish more or less 13 different groups that were correlated within the group, but the groups were not so much correlated among each other. So we kind of recommended to use one trait from each group to kind of represent the whole group itself. So for example, it makes no sense to use wing length, wing width, and the wing area if they are highly correlated with each other, but just to take one of the representative of these. This whole database is freely accessible online, and it's open to anyone who would like to use those traits for some functional trait analysis in the future. Okay, so this brings me to the second part of the study, where we actually use those traits and try to analyze how they influence the occurrence probability of the beetles under a certain conditions. For that, we gathered quite an extensive data set of almost 400 plots across Germany. These data came from 3 independent projects: the Biodiversity Exploratories, BioClim, and the Steigerwald project. So altogether, we had 5 sites in Germany, covering pretty nicely different kinds of forests in Central Europe. The sites covered unmanaged old-growth forest up to the intensively managed forest, from broadleaf forest to coniferous, and from the lowland up to the mountains. At each site, we collected beetles using the 5 intercept traps, and we also collected environmental variables for each site. For that, we used 2 different approaches. We gathered quite detailed information about the forest structure using the LiDAR data, LiDAR scanning. And for that, we get the information about the canopy density and the vegetation density, which was like in the lower part of the forest. Also as a vertical structure, which means the vertical structure of the canopy. Also the terrain roughness, which was like a slope of the terrain. And we also included elevation into the analysis, which mostly served as a proxy for climate variables because it was highly correlated with temperature and precipitation. Yeah. On top of these 5 variables, we also added 4 more that we gathered during some forest monitorings. It was the proportion of conifers at each site, and 3 variables describing the deadwood. So the deadwood amount, richness, and the deadwood structure. All these 9 ecological variables, or the forest structure variables, are known to be the most influential for saproxylic beetles. So they were not selected just by chance, but we already know from the literature that these are the most important ones. And at the same time, they were not correlated with each other. Okay, as a species traits, as I mentioned before, we used 2 different categories, some of the ecological traits and some morphological. The ecological traits we included in the analysis were the preference of species to different canopy openness and to different decay stage of the wood. Also, the information if beetles visit flowers or not. What is the preference for the host tree and the habitat? Out of the morphological trait, we decided to use these 9 traits that are in yellow, and they kind of represented 5 the most important ecological functions in beetles, which was like the coloration, body shape, locomotion, sensory, and the foraging. Yes. Okay, so to analyze the data, we use joint species distribution models. You probably all know single species distribution models, which are quite frequently used to predict the occurrences of single species under different conditions. So these joint species distribution models kind of extends this for the whole community level. These joint species, unlike the stacked species distribution model that actually works on the single species level and then pull the data together, this joint species distribution model kind of share the common responses of all species at once, which has a big advantage because they can kind of borrow the information from the less common species or from the more common species to less common species if those species share some same traits or if they are phylogenetically more similar. So we analyze the data using the HMSC package, which stands for the hierarchical modeling of species communities. And basically it is an analog to the hierarchical multivariate GLMM models, but in this case, they are fitted with the Bayesian inference. These models are quite powerful because they kind of allow to estimate the responses of individual species to environmental variables. And plus, they also estimate the influence of traits and the phylogeny on these species stages. They can also be used to identify the species co-occurrences in the samples or in the communities, but this is something that we didn't work with for now. So basically, we fitted the present absent model and used these nine covariates, 9 forest structure covariates as a fixed effect, plus 2 random effects on the level of the region and on the level of the sites as a GPS coordinates. We use the complete phylogeny for all the species that we have worked with, and we fitted 2 independent models, one for the ecological traits and one for the morphological traits. So now a little bit about the results that are somehow connected to those traits. So basically, here are 2 heat maps showing the relationships between individual traits and the forest structure variables. These came from 2 independent models, one for ecological and one for morphological traits. Basically, the red color represents the positive relationship, the white, the blue color represents the negative relationship. The darker colors are the values that are different from 0 on the level of 95% of the posterior probability. The lighter colors, there the threshold was a little bit lower to 85% of the posterior probability. So basically, let's look at the ecological traits. You can see that there are quite a lot of non-random associations, which is not very surprising because basically all these traits are somehow inherent to those species and they directly link the species with the specific resources. We can see an example, for example, here, that the occurrence probability of beetles that prefer coniferous trees decrease with the increasing canopy density and with the increasing vegetation density, and increase with the elevation and increase with the proportion of conifers, which makes totally sense. Yeah. So sometimes those results were quite trivial, but at the same time, it's a nice validation that all these ecological traits that were not measured, but just estimated usually by some beetle experts are useful and relevant. What is quite important and surprising, I would say, is that, that we didn't find many associations between the morphological traits and the forest structure variables. This is quite surprising, especially even for the body length, because body length is quite known to be associated with many ecological processes and functions, and therefore we expect it also to be kind of related to some of those forest structures. Here it's there are just some plots with the community weighted means of body size against the different variables. You see that there is no pattern at all. It is important to note that we used quite large data set of Saproxylic beetles, probably one of the largest in Germany, and also all these forest structures were, or most of them were quite detailed, coming from the LiDAR scanning. So basically, we have a couple of explanations for this. Firstly, we recorded quite strong influence of the phylogeny on species niches, which kind of means that, or which might mean that there are still some additional phylogenetically correlated traits that we did not include it in our study. So basically, could be some traits related to the physiology of the species or some additional traits. So this is like an open door for another analysis if those traits will be someday available. The second explanation for that could be also like the spatial scale of the forest variables that we collected, because it's important. There are some examples, for example, that the biodiversity of arthropods usually increased with the canopy density, but only on the like the stand scale or on the tree scale. So it means that the tree which has like a dense canopy usually provides a lot of different microhabitats, and then more species could be present there. But if you look at it on the stand scale of the whole forest, usually the canopy density decreases with the increased biodiversity. So it means that usually the open forests are usually more rich for species. So we also think that these ecological variables that we collected should be also considered in this term of the spatial scale. Yeah. So, and let's talk a little bit about the third part, and these are the bioclimatic regions, and if those patterns that we observe only for Germany are somehow consistent across the different parts. For that, we gather data from 3 independent countries, Norway, Finland, and Germany, covering different forest types, going from the temperate zone to hemiboreal and the boreal forests. Most of the data were collected and analyzed in the same way as I already talked about. But we had to change the forest structure parameters and the variables that we collected because they had to be same for all 3 countries. So we were quite limited about those. Also, we had to add a new variable, effort, that was kind of accounting for different sampling effort across these sites. Then we ran the joint species distribution models again for each country separately. And I will just show briefly some results again. This is just an example of one morphological trait that is plotted against one forest parameter, forest age. Again, the circles are the mean values of the gamma parameter and the credible interval. If the color is red, then it means that there is a positive relationship. If blue, there is a negative relationship. If it's gray, then the values didn't differ from 0 on the level of 90% of the posterior probability. So when you look at all the possible combination that we had for all 3 countries of the ecological trait, you can see this may be a little bit confusing plot. But basically, the important thing is that there is no congruent or consistent pattern across the countries. So even though we know that including the traits into the analysis can make a better prediction for the species, but those effects are not consistent across all the countries, and apparently it works differently. So just to summarize the results, so basically we have available some morphological and ecological traits for Saproxylic beetles now. And although ecological traits seems to have an ability to predict species responses to these environmental changes, we did not find any association between the morphological traits and the environmental characteristics. Also, there was probably no apparent consistency among the climatic regions, but on the other side, we found quite a strong phylogenetic signal in species niches. And this means that there might be a set of different traits that might be useful to use next time that can actually explain this variation. For example, this chemical perception or some gut microbial assemblages. Basically, all these traits should be somehow related to the species physiology. So to somehow conclude the results, we can say that probably the information we still have about this morphology or about these traits in general and saproxylic beetles is still quite limited, and unfortunately it's still very open and we cannot say much about this. Yeah, so thank you for your attention. Yeah.
You ran your analysis separately for the ecological and the morphological traits.
Mm-hmm.
Did you make any tests whether you find any effect of the morphological traits on the ecological traits? Like, because looks like the morphological traits, they are not influencing anything.
Yeah.
But so they are not a good way to describe the ecological trait, right?
No, they are not a good way to use it to describe the effect towards the species niches, which means towards these ecological variables that we collect.
Yeah, I was just wondering if they are also not even able to, like, because when you showed that matrix with the correlations, the first one, you were saying that those groups of traits, they probably had a role for either diet or something, but then you showed that they don't have any effect. So do you think they really have no effect on the ecological traits? Or it's just that they are not affecting enough?
Yeah, well, at the beginning we thought that there will be some connections between the trait and the ecological variables, that we can actually link the trait with the core structure parameters. But well, at least in the way that we analyze the data, because there are different ways how the data can be analyzed and different approaches and everything. So in the way that we did it, unfortunately, we couldn't observe almost anything. So
related to this, I think first I think it was a good point to do the morphology versus ecology species traits. That there could be some results.
Well, the thing is that these ecological traits are very general, and it means when you think about it, that they usually cover different morphological traits. For example, if the beetle, prefers coniferous forests, so it means that morphological there can be many morphological details that can go with this. So that's another way why we shouldn't probably call these ecological characteristics traits, because they are not like the traits in the strict sense of way, and they are mostly representing a broad range of different traits itself, like physiological, morphological, or those. So I don't think that the direct connection of these ecological morphological traits would make a lot of sense, actually.
Yeah, they would have to be more precise, like whole specialization would have to be somehow quantified, and so on. Could you go to that heat map of the morphological correlations? No, the big one with morphological traits correlated. You said you extracted effect of body size, but when you look at it, it's all blue. So normally you would expect that some will be half will be red, half will be blue, basically. But somehow everything goes positively together. Is it not that there are some traces of the body size? There's a signal of body size yet, or how would you explain that?
That's a good question. I don't know. I didn't think about it that everything goes in the same direction. That's true. But
everything is either getting bigger or smaller. So I suspect this is basically still some, something to do with body size.
Yeah, well, with the body size is a problem to work with all these measurements because the body size is so prevalent, so it goes to all these traits that we measure.
You can have different allometries for different traits. Yeah. So if you although most of your traits are linear measurements,
that was the problem. Another.
And then last question on that, you picked up few of them and did correlation. Did you consider to extract some, PCR variables?
PCA.
PCA variables instead, so that you have sort of synthetic.
You mean to work with the canonical axis and the eigenvalues with those? We did PCA at the beginning, but then we kind of switched to this approach mostly, and we didn't try to work with those. But we try to somehow distinguish between different morphological traits, but just based on this PCA at the beginning. Yeah, that's true, but we didn't summarize the results across them.
I don't necessarily like that approach because then you have, even if you have nice correlation, you don't know what exactly it means. If you have, complex PCA axis, but sometimes
this is in general quite complicated how to get rid of the effect of this body size because it's so prevalent and it's everywhere, and especially as you pointed out, some of those traits are not linear like body mass, and so they grow exponentially unlike the others, and so it's a little bit tricky to work with it. So we decided at the end to use these residuals from this phylogenetically correlated regression model, which is one way. We discussed this with different people, and different people have different opinion about the same situation, and we still are not sure if this is the best solution for it, but it's one of the ways that we decided to go.
It might be the reason why you are not getting body size effects and morphological effects on distribution, because if you take out, body size is phylogenetically very correlated, and so if you get, if you get it out of that, which has a merit of, you would read, your measurements as independent phylogenetically, but at the same time, when you look at the function of the forest, the function of the body size in the immediate community has nothing to do with what's the history of that body size. And so, at least for some analysis, I would ignore phylogeny and look simply whether, bigger beetles are doing something different than small beetles, and completely like functionally completely ignoring the historical issues here. Maybe
could be, yeah.
So, great. I have So, if I understand it correctly, you didn't find much evidence for some habitat filtering in terms of the morphological traits.
Yes.
Because the beetles living under similar conditions did not show similar traits.
Yes.
Just out of curiosity, did you test the opposite hypothesis, that the beetles occurring under similar conditions will be less similar than you would expect based on their relatedness? So instead of having some clustering and habitat filtering, you would have over dispersion in the community.
We did not test this, but I'm not sure how to test it in the way.
We can discuss it. Botanists do this.
Well, basically, we didn't try this.
What's the data you used from the biodiversity exploratories based on one collection that was recently done, or did you also incorporate older data into that?
Well, to be honest, the data was given to me, so I'm not an expert of these biodiversity exploratories, and I know that these data were collected in different years.
Okay, yeah.
So I have no idea. I would have to check what year was the data from.
Yeah, no worry. Just
like 2007, but I'm not.
Yeah, I just know there's a lot of data from the exploratories.
Yeah, I know, and it's still going on probably, and they're still adding new data. But yeah, it was like the most recent and available at that time that we worked with those data.
Gosh, so if I understand correctly, like the final result where the morphological traits didn't have influence, they are like already based on this model where the body size is filtered out on log scale, but also phylogenetical relationship. Like these results are already after correcting.
Yeah, after correction, because otherwise all the traits would be correlated with each other, and there would be no point of adding all different traits in the same analysis, because they would show all
because as what you said, like I think also, like, you know, especially in beetles, if I imagine them, a lot of these like morphological traits will be highly phylogenetically conserved. So if you filter that variability out, maybe you just filter out all these like kind of influences of morphological traits. Maybe, I don't know. There are like different ways like to do it. Like we are doing a little bit similar analysis now on ants using only morphological traits because we don't have any better as well. And we are testing rather what Martin described, like, you know, this community over dispersion and clustering. And there we can see actually still really interesting patterns, even if you filter out the file.
And you work with this,
but it's a different approach. It's like a null model where within small site, you are like kind of assuming, you know, there are these null models of species co-occurrences, and you can run something similar with traits, so asking if the co-occurrence of similar or different traits is like clustered or over dispersed. So I think it's a bit different approach than direct correlation on Bayesian scale of different variables. But I can imagine if you are interested.
But it would be nice to try this as well for this data.
Yeah, of course. So depending on question and on the scale, like, because as you described, maybe single tree in terms of species coexistence is something else than the broader habitat. And
do you see any results, like any connection between the traits, like morphological in ants?
Like the pattern we saw, like we got, for example, over dispersion in the communities in traits, and we saw it even stronger after we filter out the phylogeny, which was kind of interesting. But it's a different approach. It's like from Francesco de Bello, where you can decouple the phylogenetic and functional metrics, like, you know, at the start. So I think it's a bit different than what you are doing here.
And also inform, you know, like on the relative importance of like abiotic factors versus symbiotic interactions for linear communities, because I assume Peter is seeing over dispersion because the ants are competing.
That's what we think. We actually got opposite result than we expect. Before the primary forest will be still functionally more diverse. But once we start the analysis, we got surprisingly switch, and the functional, like over dispersion of traits was much stronger in ugly secondary forest, which we think could be competition, but we cannot be sure because this advantage of these new models is that we are interpreting just the pattern, and now many studies showed that, for example, over dispersion can still mean competition and niche filtering depending on history, situation, interactions of species. So it's like there is so many critics now that you cannot be certain just from the pattern what's the mechanism. But you can get definitely interesting patterns even with morphological traits when you filter out phylogeny in our case. But here for your broader question, I was also wondering that maybe the morphological traits didn't have effect because once you filter out phylogeny in beetles across so many lineages, they might be so highly phylogenetically conserved that maybe that's the reason.
Could be, I don't know.
Yeah, but very interesting.
That's good. Very interesting. We can. But the problem is that once we not decide to filter out the effect of the phylogeny, then we probably are not able to work with the residual values of these interaction of the measurement versus the body size. And if we are not able to do this, then all the measurements will be highly correlated with each other. So
you can do residuals on body size only.
Yeah, I know, but this is some. Yeah.
Sorry, I have to go examine students.
Yeah, but something that you are not exactly suggested to do that. Right? That actually people told us that without the accounting for the phylogeny, it makes no sense to do this. But
it depends on the research, because I know that Francesco they were always comparing, like 10 years ago, already comparing the analysis with phylogeny inside and without, and each of them was a bit different.
okay.
It was fine.